Here in part 3, let's talk about photosynthetic diversity and a process known as photorespiration. Guideline number 5. C3, C4, and CAM represent 3 distinct photosynthetic pathways, and there are important distinguishing features in them associated with biochemistry, with phylogeny, and with the carbon isotope ratio value that we detect. Here you see in simple diagrammatic fashion what we expect to see for the anatomy of a C3 plant, of a C4 plant with its Krantz anatomy in the center, and in CAM plants. Now there are large differences in 13C between C3 and C4 pathways, and we find that C4 plants and CAM plants tend to have about the same value. C3 plants represent about 95% of all of the plants on the earth today, yet they only account for 70 to 75% of primary productivity. C4 plants are less than 5% of all the species on our planet, yet they account for about 25 to 30% of all terrestrial. CAM plants are less than 1% of all the plant species, and they account for less than 1% of terrestrial productivity. There are 2 primary groups in angiosperms that contain the C3 and C4 species. Monocots have about 12,000 species, and about half them are C3 and half are C4. Dicots represent about 300,000 species, but only 2,000 have C4 photosynthesis. An important and distinguishing feature associated with C4 photosynthesis and the abundance in the monocots is the notion of parallel veinal structure in monocots versus the reticular or web-like structure in dicots. C4 plants are found in the more derived angiosperms. They appear in several families, and within those families they appear in several genera. And evolutionary studies tell us that C4 photosynthesis might have independently evolved multiple times, even within the same genus. CAM plants are distinct. Typically they're succulents. CAM stands for crassulacean acid metabolism because it was first discovered in the family, the Crassulaceae. But it is quite common in the Cactaceae, and in the Agaveaceae, and in the Liliaceae. CAM is present in ferns, gymnosperms, and angiosperms. Its independent evolution is also a commonly observed phenomenon. Here you see a number of the different families in which CAM has evolved. The most common taxa for CAM are the bromeliads and the orchids, and they're primarily in arid lands and in tropical regions. A large number of the dicots occur in arid habitats on a global basis. So what is the difference between C3 plants and C4 plants? In C3 plants, CO2 comes into a plant. The enzyme RUBP, the substrate RUBP, and CO2 are combined through an enzyme known as ribulose bisphosphate carboxylase, short term RUBP carboxylase, or Rubisco. A 3-carbon molecule is formed that's stable, that is a 5 and a 1 form 2 3s, hence the name C3 photosynthesis. Then we have the photosynthetic cycle, spinning off sugars, reproducing RUBP to continue the cycle. In C4 plants, the same process occurs, except it is now confined to the interior of the plant, typically in a bundle sheath cell, such as shown here in this cross-section of a grass. In the outer cells, a second process takes place, and that is that CO2 combines with phosphoenol pyruvate to form a C4 acid. That's the first stable product, hence the name C4. The C4 acid diffuses into the interior cells through plasmodesmata openings, where it's decarboxylated, releasing the CO2, and pyruvate comes back. That pyruvate has to be regenerated into phosphoenol pyruvate, which is making anaerobic respiration go backwards, and then we can repeat the same cycle. CO2 that comes in to the bundle C cells does not need to remain there. It can diffuse back out. But if it diffuses back out, then it is refixed again by PEP carboxylase, so that essentially CO2 never comes out. And the consequence is that since RUB carboxylase is slow and PEP carboxylase is fast, that this acts as a CO2 pump, resulting in high CO2 concentrations. Now, the cycle I've been describing so far is a photosynthetic carbon reduction cycle. That is, CO2 molecules combine with RBP through rubisco and form PGAs. And for 10 cranks of the cycle, 10 carbon molecules are fixed as organic matter. That's what happens in an environment in which we have 2,000 ppm CO2 and 21% oxygen, similar to the environment we think existed about 10 million years ago. Today is a very different environment. We still have 21% oxygen, but we have about only 415 parts per million CO2. And in that case, we discover that RUBP carboxylase actually has a second function. That is, the CO2 can combine with oxygen or with carbon dioxide. If it combines with oxygen, we call that PCO, photosynthetic carbon oxidation, versus photosynthetic carbon reduction. When it combines with oxygen, we have phosphoglycolate formed instead of phosphoglyceric acid, and that eventually gets shuttled around, resulting in the release of carbon dioxide. This process of releasing CO2 during photosynthesis is known as photorespiration. It only occurs during photosynthetic periods, and any process that results in the emission of CO2 or efflux of CO2 is known as a respiration. The consequence of this atmospheric composition today is that for every 10 RUPP molecules, we find that we have 54 PGA, not 60. So we have net carbon fixation of only 7. So there's a reduction in the efficiency of the reaction in today's environment. This photosynthetic process and photorespiration are both a function of temperature. And it turns out that photorespiration is directly related to the CO2 to O2 concentration, and it's directly related to temperature. So that as CO2 concentrations decrease, we have an increase in photorespiration relative to photosynthesis. And as temperatures increase, we also find an increase in photorespiration. So the same active site in ribulose bisphosphate carboxylase that has also this oxygenase function is sensitive to both CO2 concentration and to temperature. Guideline number 6. C4 and CAM plants have similar carbon isotope ratios, but they don't have a single value. The C4 variations reflect bundle sheath cell leakiness. I apologize, I have a coarse voice today. And CAM variations reflect a mix of C3 and C4 CAM-like activities. CAM differs from C4 photosynthesis in a very simple way. The two carboxylase reactions that are separated in C4 plants actually occur in the same cell in CAM plants. To prevent these two carboxylase reactions from competing with each other, PEP carboxylase is active at night and RUBP carboxylase is active during the day. The C4 acid doesn't diffuse into another cell. It's instead stored within a vacuole.